Next slide, if there is one. Our voice, our newsletter. If you haven't taken one, please take it if you're interested in it, if you want to subscribe to it. It's free electronically. If you want to get the written, printed version, there's some mailing costs involved. Pretty unique. I think you will enjoy it. And this is something new that some of you may not be aware of. We launched a new website called the Reproductive Times. And the idea there is to provide information on reproductive medicine and basic reproductive sciences. It's kind of an electronic newspaper. We're posting now 2 to 3 times a week, several articles. We're looking for people to collaborate. We're interested in materials. If you have something to say, let us know. We would love to have you participate. Again, the idea is for people to have a central address to find out what is happening in the field scientifically as well as socially and in every other aspect. Okay? Which brings me to today's presentation. Our topic is in vitro manipulation of ovarian follicles and endometrial organoids for bench-to-bedside research. This is a topic chosen by our speaker, Shuo Xiao. You see his qualifications here as a tenured associate professor here in town. And I invited him because I read the recent one of his papers, and I found it quite interesting. And so he is going to, I think, give us some very interesting information on follicles and endometrial organoids. Pleasure having you.
Thank you. All right. Good evening, everyone. At first, I would like to thank Dr. Glacier and also CHR for inviting me. Am I good on time? Okay. For inviting me to be here. And when I came to this floor, I realized, oh, this setting is very different from what I used to do a presentation, but it's very nice. I mean, the nicer way. So it's very casual and also very fancy too. So by the way, my name is Shuo Xiao, and from my accent, you may know I'm not from here. Actually, I was born in China, and I went to medical school and in China too, and then I came to United States for my PhD, and then I primarily focus on research. So today I will talk about how we use the in vitro reproductive tissue or automated culture model. So for us to understand reproductive primary female, including reproductive biology, toxicology, medicine, and also disease too. And in the last 2 years, I'm also more interested in drug development, especially the non-hormonal contraception, and also some potential drug target for treating PCOS. So I will talk a little bit about that. So just give you a very general introduction about my life. And so I was born in Beijing, in China, the capital city in China, and right here is the forbidden city, but I was not fortunate to born here, of course. This is for the I try to. Yeah, actually, my home is a small apartment right there. Yeah, and then in China, we have different medical school systems, so we can go to medical school right after the high school. So after the high school, we passed very competitive the test, and then I went to Peking University and finished I just call either Bachelor of Medicine or MBBS. So I spent 7 years there, and then after that, I did 2 more years for my public health degree. I focused on women's breast cancer and also toxicology. And then 2008, I came to University of Georgia and started my PhD. And during my PhD, I didn't focus on the ovary, I focused on the uterus, especially the uterine receptivity and the embryo implantation. And then after getting my PhD, in 2013 I went to Chicago and joined Dr. Teresa Woodruff lab for my postdoc research. So a lot of you may already know Dr. Teresa Woodruff. She's a pioneer in onco-fertility, and she contributed a lot.
She gave us her GrandRonds.
Oh, that's great. Yeah,
Yeah,
it's
in
an
this
honor
room.
to follow her way. Yeah, cool. Yeah, I did my 3 years postdoc with Teresa, and we still talk a lot. I just met her in Dublin during the SSR meeting 2 months ago. So in 2017, I started my own lab in University of South Carolina, and then I primarily in School of Public Health, and then I primarily focus on environmental and reproductive toxicology. We want to study how endocrine disruptors will impact females' ovary, uterus, and overall fertility and their hormone secretion. And then I realized, I want to move to the north, and especially at Rutgers, we have one of the best toxicology program, because toxicology was one of my major focuses. So I moved to Rutgers, in School of Pharmacy about 4 years ago. And when I moved to Rutgers, I thought I will come to New York City very often because it's so close. But actually, I just told some of my new friend I know today, this is my third time actually. Yeah, in the past 4 years, because it's a little bit complicated. Yeah. So, but I made it today. So I dropped to Princeton Junction and tried to take the train, but then I realized, oh, my train was canceled. So I called Uber and then spent 2 hours get here. So overall, that's I believe you right now you know me better, and just call me Shuo. Yeah. So for the next part, we can talk more about our research. So right now we primarily focus on female side, especially the reproductive health. And you may realize for my PhD, I focus on the uterus, and during my postdoc research and in the past 6 years of my faculty research, I primarily focus on the ovary. And right now I have more motivation to engineer the female reproductive system on chip by integrating both my expertise in both ovary and the uterus. So I believe most of people already know human reproduction very well. So usually this slide is for a toxicology conference, so I can save a lot of time on this. But overall, I believe everybody agree with me the ovary is very important, because we focus on the ovary, the uterine implantation, no matter you focus on research or you are the REI right here in this room. And we also know reproductive disease are pretty common. It's a big issue all over the world, including the United States and also where I was born in China. In China, actually, we see a lot of more rate of the infertility, including male factor and also female factor to either nutrition or environmental pollution. So that's one of the reasons I primarily focus on how environment will impact female reproduction too. And since I primarily focus on toxicology, we are also interested in how environmental toxicant, how chemo drug, irradiation, and even the emerging immunotherapy will affect women's reproductive health, especially in the ovary, and we do some uterine research as well. So make the long story short, so currently my lab is primarily focused on this 4 areas. So currently we focus a lot. We have a relatively big lab. I didn't expect, we got a lot of grants in the past 4 years. So right now we have about 8 to 10 postdocs and then several students, almost more than 20. So if for academic lab, whenever you have more than 10 people, that's a lot, pretty overwhelmed, yeah, for me. So overall, we focus on female reproductive toxicology and also biology, because I always believe biology is the key for us to do toxicology research. And we also focus on the fertile protectant development, especially the continuation of the onco-fertility research after I was trained with Teresa Woodruff. So we want to understand how chemo drug radiation will kill young female cancer patients, the ovary, especially their germ cell oocyte, or how they diminish their ovarian reserve or exhaust all the primordial follicle. And I specifically want to focus on the damaging signal pathway and where is a unique druggable target, so we can develop a fertile protectant without affecting the efficacy of the chemo drug. and then they don't have to remove their ovary, but still preserve their fertility and also the endocrine function. And then when I was did my postdoc with Teresa, you may heard she is also one of the pioneer people doing the microfluidic system and the organ on chip, especially the menstrual cycle on chip and also female reproductive system on chip. And when I read her paper, actually I used to lead the project. So at that time, I started to culture human follicle, mouse follicle, and also other tissue organoid, and build the female reproductive system on chip to enable us to study their biology, medicine, and also toxicology as well. So since we can culture follicles, we can culture uterine endometrial organoid and build a Melinda Gates Foundation. Also, collaborate with us, and we do a lot of ovulation screening using our organoid model, and then we try to develop non-hormonal contraception. So I will bring this a little bit, and then primarily focus on the reproductive medicine and disease-related topic, since most of the audience here, you primarily focus on the clinical side. So why we need the organoid organ on chip research? So for us, we always think about whether we have for example, the stem cell induced organoid, whether it's ready for basic science or especially for toxicology research, whether it is representative enough for human ovarian function and especially for the oocyte development process. But I will give you it's open for discussion. So overall, currently the gold standard in research field for studying reproductive biology is still using the whole animal models, especially in the academic side, we use a lot of mice, but actually mice are not human. We may all agree, actually there's a species difference, even though most of the reproductive biology is relatively well conserved in mice. We find a lot of new novel discoveries to advance our understanding on reproductive biology and also medicine. However, so one disadvantage of the animal model is relatively expensive, even though compared to human study or clinical trial is still it's much cheaper compared to clinical trial, but it's for us, for researchers, it's still very expensive. For example, we pay about $10,000 on our mouse breeding colony and every month actually. And also for another reason we want to have the organoid or in vitro model for studying ovary and uterus also because reproductive events is pretty dynamic. I think everybody already understand actually for folliculogenesis or follicle maturation process is pretty dynamic. So how we can culture primordial follicles as possible, especially for human follicles. Overall, in vivo, it takes more than 200 days for a primordial follicle to go through the whole process, and then it can become ready for ovulation. So can you imagine if we can culture any cell on a dish, for 200 days, whether that is possible, right? And also for hormone secretion, ovulation, and those processes are also pretty much dynamic. So if you want to study the follicle development, and either by their size or their hormone secretion or the ovulation process, in human or in pet mice, it's almost impossible if we do not sacrifice mice. So we have to kill a lot of mice and then study those dynamic process. And actually in the toxicology field, or the US EPA, the Environmental Protection, or the FDA, so there's a new word coming up this year. It's called NAM. It's a new a methodology or new approach or methodology. So they really want us to ensure the animal welfare, so we can reduce the use of animals and also avoid the species difference. And of course, the uterine decentralization or implantation process is also very dynamic. If we do not sacrifice the animals, it's really hard for us to do it. So actually, one of the PhD students, I go to animal room every morning to check the vaginal plug because I don't want to miss any mice. Whenever I miss the plug, and then this mice cannot be used. So even including a Thanksgiving and also the Christmas Eve time. So and also So and also there are there are also some human primary cell lines. For example, in IVF clinic, we can get red luteinized granulosa cells, and then we culture and we can reprogram them back, and they can still express some follicle maturation genes, but overall they are not super perfectly representative to what is happening in vivo. For example, so for a follicle, we can just treat follicle as a mini ovary. Okay, so actually for the ovarian sterogenesis process, hormone secretion, and also oocyte maturation process, they need different cell types to work together to support the follicle, like development, maturation, and also ovulation too. So, for example, the theca cell in the outer layer, they primarily produce androgen by responding to the RH, and also some people claim IGF-1 is very important too, but they don't produce estradiol because they don't have aromatase. So the inner layer of the granulosa cell, they have aromatase. They will take the androgen and convert the androgen to estradiol. So there's something wrong, and then they may cause PCOS or follicle development arrest, and then infertility or other reproductive disease. And that's the somatic cell communication. So actually, those somatic cells, they also talk to the germ cell oocyte. For example, the oocyte, they can secrete GDF9 or BMP15, those TGF-beta family proteins, and then they can drive some granulosa cell to become cumular cell. And cumular cell is very different from murogranulosa cell. They have their own function. They protect the oocyte. They will be ovulated with the oocyte together in the fallopian tube, and then play a very important role in like fertilization. So and also, so actually granulosa cell, they also very important for supporting the oocyte health. For example, there are some gap junction or TZP transposal projection, so they can provide nutrient to support the oocyte maturation. So for example, the oocyte, they don't produce amino acid, pyruvate, and also cholesterol. All of them, they need to be from the somatic cells. So probably you can appreciate actually if you want to do the IBM, in vitro maturation, and only if you culture oocyte only, it won't work, right? And also if you culture COC, cumular cell oocyte complex, you may expect actually the cumular cell, they may differentiate to some murogranulosa cell like cell type 2 because they miss the support from the murogranulosa cell and also the theca cell. So based on all these facts, we may want to develop the organoid or in vitro model so to enable us or provide us with an additional option so we can study the reproductive biology, disease, and also medicine as well. So actually, I started to learn this technique in 2013 when I did my postdoc with Teresa Woodruff. So at that time, probably you may heard about her technique. So we use hydrogel or alginate hydrogel to encapsulate either mouse or human follicle to give them a 3D environment. And then they are pretty happy, they can grow, and then we can give them FSH, and then they can develop from the, for example, the secondary stage on day 0, if we culture them for 8 days, they can grow to early antral stage on day 4, and then to the fully mature antral stage on day 8. And then on day 8, if we give them RH or HCG, the RH analog, they can respond and ovulate. So you can see the follicle, they can rupture on one side, only one side. They can mimic what is happening in vivo, and then they can ovulate the egg with a parabolic extrusion. And after ovulation, the remaining somatic cell, they can also luteinize and secrete a lot of progesterone. So if you see the ELISA data right here, so overall the follicle, they can secrete more and more estradiol to mimic the follicular phase or the proliferatory phase if you want to see the uterus. And then here is the ovulation time, And then after ovulation, the corpus luteum, or we call it corpus luteum spheroid, they'll secrete more progesterone. So overall, this pattern can mimic what is happening in vivo. And also for the ovulated oocyte, you can also perform IVF to primarily by biopsy. So because what we found is, so when you culture follicles, the donor pellucida will become pretty hard. So if you do traditional IVF, the rate is pretty low. But if you do ICSI, inject the sperm, and then they can become 2-cell, 4-cell until morula stage. And then if you transfer this embryo, you can make the pseudo-pregnant mice pregnant, deliver laptop. But I want to be honest, actually, if you culture follicles on endocrine level, it's pretty happy, well controlled. But actually, for the oocyte, the success rate is only 20%. So that means actually the media, the recipe is still not as what we have like inside the body. So we still need to optimize the culture condition. So for here, I have like two videos. The first video is about 48 hours, like a follicle culture time, and you can during the FSH stimulation, when you stimulate the follicle, you can see actually the granulosa cell on the like outer layer they keep proliferating. Now actually the oocyte you see, I don't know. Actually, I still don't know what is happening in the oocyte. It's something there. It's changing. Partly because they produce a lot of proteins, maternal effect factor, and they also increase in size to become fully mature. And then after 8 days culture for the antral follicle or the fully mature follicle, we give them HCG, and you see what happening like during the 14 hours. Because it's a mouse follicle, the ovulation time is about 14 hours. You see, it's very interesting that actually only one side follicle is rupturing, and then they try to push the egg out. And then if you add hyaluronidase and get the single oocyte, actually the oocyte right here, and you see the oocyte with the polar body extrusion is pretty nice oocyte. Is comparable to if you do the super ovulation and get the oocyte from mice actually. So and then after I have my own lab, we also want to know so whether this cool model can also preserve the FSH or LH like a dependent follicle maturation and ovulation process like at molecular level, because we really want to use this model to go like a deep dive and study like a drug or target or toxicology. We want to know the molecular mechanism. So we also did a lot of single follicle RNA sequencing like a proteomics and also single cell RNA sequencing to compare what we found in vitro and with the in vivo data. So this is a busy slide, and we have many, like several papers, like showing that the answer is yes. Actually, relatively, it's pretty much like physically conserve what is happening in vivo at molecular level too. For example, for the two cartoons on the top, that is what we summarize the key signal pathway and during the follicle maturation and also ovulation. For example, the maturation is pretty much FSH dependent, even though RH is important. But right now, I believe RH is good, but it's not truly necessary. If you don't have RH, that's fine. For example, in IVF clinic, you don't give them HCG, or you can give them mild HCG. But if you only give them FSH, so the follicle can still reach the full maturation. We're open for discussion. And then during the ovulation, that is pretty much RH or HCG mediated through the G protein coupled receptor. It's also called RH receptor. It's pretty complex, and you can see I just have some example. For example, so in our model, so actually the 3D culture model or the mini ovary model, so we can see if we culture them for 8 days, actually the FSH can hugely induce the CYP19A1 or the aromatase, what it is for like converting the androgen to estrogen, right? And then the follicle can produce more and more estradiol. And then we also see a lot more RH receptor. The gene's name is RHCGR. And actually, initially the RH receptor is only in theca cell for androgen production, but later on it has to be very abundant in granulosa cell because it can be ready for RH surge for ovulation. So we also see a lot of induction of the RH receptor in the granulosa cells. And then during the ovulation time, you see this is about 0, 1, 4, and 8 hours after the HCG treatment. And for human, you can just double the hours because for human ovulation is about 36 to 48 hours. So we see a huge induction of the progesterone receptor and also the AREG. So actually, progesterone receptor is very important for follicle rupture. So if you delete the progesterone receptor, the follicle cannot rupture, cannot push the oocyte out. And also, EREG is the epidermal growth factor receptor ligand. It's also very important for the ovulation process, especially the cumulative cell expansion and also the oocyte meiosis. So overall, if you are interested, you can read our papers and get more detail. We have the full gene list in our supplementary table. So in addition, so all the data are based on mouse follicle. It's really because the stem cell induced follicle, no matter granulosa cell, theca cell, or the oocyte, is still not ready for doing the in-depth biology. But actually, when I was in Chicago, I also used the same method to culture human follicles from the National Physician Cooperative. but probably through the oncofertility program. So whenever young female cancer patient, they want to preserve their fertility, so they will do the surgery and remove their ovary, and then 20% of the ovary I am the point of contact in New Jersey, in Chicago to receive the ovary, and then I used to isolate about 56 secondary follicles, and then I use the same method to culture them, and it takes much longer time. Usually we have to culture them for about 30 to 40 days. And also the successful rate is pretty low. You see, we only get 4 M2 oocyte, but that's still the first M2 oocyte ever from the cultured follicles. But success rate is only 10% actually. So we did some marker and see whether how about the quality of the oocyte, but we cannot do that because this project is funded by NIH. We cannot fertilize the oocyte unless the private foundation budget is very strict. So only spend some marker. We know that we have the polar body extrusion, and maybe you have better idea, based on the morphology, whether it looks okay or not. But actually, we didn't do that. We didn't do ICSI because it's not allowed. So overall, that's I hope you agree with me. So actually, the 3D follicle culture model, especially based on the hydrogel encapsulation, they can rapidly okay, and they can recapitulate or mimic what is happening in vivo at a hormone level, morphological level, and also molecular level as well. And actually, for me, I'm more interested in applied science or how we can use this model to truly advance the women's reproductive health. So we want to ask the question whether we can use this model to study the biology for deeper understanding, and also whether we can use this pool model to screen the endocrine disruptor and other toxic drug and for their toxic mechanism, and also see the protective or preventive method. And we are also interested in some ovary disease, for example, the follicle development arrest, or recently we are interested in PCOS. So we also manipulate this model to mimic the PCOS ovary. We are not ambitious to cure PCOS because it's so complicated about endocrine, liver, and many other organ systems. But we want to know whether we can find some druggable target and have the REI in this room to have better ovulation rate and also better follicle maturation and IVF successful rate. So I will touch that a little bit. And we are also interested in drug development. But overall, for all this application, I have we have 3 major focus right now. So for example, as I already mentioned most of them. So for example, we are still doing toxicology work a lot. So we are interested in environmental contaminants, for example, endocrine disruptors, heavy metals, and other stuff. And we are also interested in drugs, whether they are toxic to human reproduction, especially the early pregnancy, and also what is happening in the ovary. And you never know, actually, a lot of compounds they are developed not targeting those nuclear receptors in the ovary at all, but based on NIH high throughput screening, for example, hydrocortisone is very strong progesterone receptor antagonist. And I was so surprised after I read my collaborators' data. So we are going to test that in our model too. So for women who take hydrocortisone, they may have early pregnancy loss because their uterus might not be very receptive. And then I mentioned, so Bill and Melinda Gates Foundation also found us to do non-hormonal contraception. And we also study the female reproductive system disease. And also our long-term goal is try to make an IVF on a chip. And then whether we can use it for patients. And we also do vitrification method. Everyone knows vitrification, especially embryologists. And actually, we also want to know whether we can vitrify the organoid, because comparing to vitrify the egg, the bigger the tissue or the more complex structure of the tissue is much, much harder because they have a connection, gap junction and intercellular connection. Whenever you freeze them, you break the connection and then they will die. So just a very quick introduction. For example, for toxicology work, we screen PFAS, for every chemical, and also extreme heat and wildfire smoke in New York. For example, last year in Quebec, the Canada wildfire come to New York, right? In New Jersey too. And actually, there's some rumor that IVF success rate might be lower during that event, especially it happened in Boston whenever they extreme extreme heat and also the air pollution as well. So in California, it's the same. So a lot of environmental epidemiologists study the association between wildfire smoke and also the IVF successful rate. I don't know whether because before they visit IVF clinic or some stuff they penetrate and go to the incubator. That's for open for discussion too. So, and for our vitrification project, just one slide and I will quickly go through. So I mentioned vitrified oocyte is relatively like very standard. It works so well, but actually if you want to vitrify organoid or follicle for either fertility IVF purpose or for our toxicology, like a biobank purpose, is a totally different story because if you don't have good recipe, you break the gap junction between those cells and also between the connection between the oocyte and somatic cells, and then the follicle cannot survive. It cannot be cultured. So we collaborate with Dr. Mary Zaniski from Oregon and develop a closed vitrification system. And so far it works pretty well in mouse follicle. So because I don't have like a human follicle from premenopause ovary to do the research, that's very, very challenging. So, but overall for mouse follicle, they are very, very comparable at morphological level, meiosis level, hormone secretion level, and we also did the single oocyte RNA sequencing, follicle sequencing, and they are very, very comparable. So that means this vitrification method is like works pretty well. So if you are interested, I'm happy to share all the protocol with you. And if vitrified follicle might be like able to provide an additional option for IVF or fertility preservation. And then for our non-hormonal contraception, like a contraception work, yeah, we have a whole team like working on the high throughput ovulation screening, and then we try to see whether we can see any druggable target that can be like repurposed as a non-hormonal contraception, because hormonal contraception it works well, but a lot of people for unknown reason they are very sensitive. They either have obesity, stroke, mood issue, and even some cancer risk. So for the next 10 minutes, I'll just briefly introduce how we use those culture model to learn more, or deep understand the reproductive disease biology and also the more related discipline about the IVF. So for this one, I want to primarily focus on FSH. So I believe FSH is the major focus in IVF clinic because we want to mature the follicle, we do the ovarian stimulation. Primarily, we want to mimic what is happening about the gonadotropin dependent follicle maturation process. So everybody already know those, I don't have to mention that. For what we want to claim is, so for FSH, because this idea is from Dr. Nataki Douglas, nobody may already know her. She mentioned she visited here before too. So whether this is FSH threshold, so whether the ovarian stimulation procedure we use in IVF clinic is too high, too low, or is not sufficient, or whether different people they have different response, I believe so. So whether we can have the personalized or individualized ovarian stimulation protocol. I heard some are they used to think about it, but it's more complicated. So right now they still follow the same protocol. So what we did is we use our follicle culture model and give them different FSH and see whether there's a threshold that is required for the follicle to acquire the dominancy or the maturation, and then they can go through the whole process and become ready for ovulation. So overall, the answer is yes. For example, in our follicle culture system, we give them different concentration of the FSH and see what is happening. So actually, in our culture system, we give them 5, 7, 8, 10 milli-IU per ml. And then from the figure, you can see, so actually 5 didn't do anything. So follicle, they can grow. The stem cell proliferation is less dependent. Overall, they don't secrete estrogen very much. And we don't see increase of the estrogen production at all. And also, but after we increase to 7 and 8, some follicle will be boosted. You see, some follicle they can have a high E2 secretion state. And then if we keep increasing the dose to 10, you see it's more homogeneous or more consistent. Actually, that means probably the FSH threshold is around 7 to 8 milli-IU per ml, but in human that will be different. The unit will be, or number will be very different. But overall, that may can explain actually for human wife, they are mono-ovulatory, where only one follicle will be selected for dominancy. So actually primarily because so only the most sensitive follicle will be picked, and then due to the negative feedback mechanism, the FSH will go down, and then the other subordinate follicle, they will go for atresia because they don't have sufficient FSH. And we just recently have a review paper discussing that, but it's so hard to convince I believe the monkey people to accept our idea. Yeah, I will keep trying. So for this data, we demonstrate there's a FSH threshold to require for stimulate the follicle maturation. And then we also want to ask if we give them too much FSH, whether it's good or bad. I think there are a lot of studies showing that might be not perfect. We want to reduce a little bit, but we want to study what is happening at the molecular level and also give us more evidence to claim this. So what we did is we give them a 10, 20, and 30. So 10 is kind of mild level. They are not very strong. And the 20 and 30, we consider them as excessive level. And then you can see based on, actually the size didn't increase a lot. But if you see actually from 10 to 20, actually they produce a lot of more estradiol. So maybe that is the reason to cause OHSS, ovarian hyperstimulation syndrome. And more interestingly, we found before ovulation, we found actually if you give them a lot of more FSH, you pretty much elevate the progesterone secretion. And this is not a new idea, it's not what we found, because premature luteinization is a standard. You will measure the progesterone level and determine whether you want to do fresh or frozen embryo transfer. Correct me if I'm wrong. So, but we also found actually, surprisingly, we found if we give them a lot of FSH, they will have very high testosterone production. And we didn't find any literature. Maybe it's we know this between REI, but we didn't find any literature regarding this. So whether we want to change the concept or the word of the premature luteinization. So whether the high testosterone will do something for the endometrium, make it less receptive. So that's why we may also want to choose the frozen embryo transfer. But we don't have data yet. But that is what we found. We found high testosterone or androgen production as well. So maybe you can measure the testosterone. We want to determine the premature luteinization as well. So just some gene expression data. Actually, so all the gene expression can explain the phenotype or the hormone expression we can we observe. So overall, what we found actually for all the genes that are related to hormone secretion, they are very sensitive. They follow the dose-response relationship. If you give them more FSH, you will have more a star CB11A1 and a CB17A1 in a thicker cell. So that may explain why we have a lot more testosterone and also progesterone. But actually for FSH receptor and RH receptor, they don't follow that. There might be a saturation effect. So even though you give it a lot more, they are okay. So, and then we want to see the ovulation outcome to mimic whether ovarian stimulation will affect the ovulation. So overall, the answer is no. So if you give them a lot of excessive FSH, actually you don't affect or adversely affect the ovulation outcome. For example, we see that when the FSH concentration is below the threshold, they don't ovulate very well. But whenever you give them more, and then you don't see a very much difference. That means whenever it across the threshold, most of the follicle, they can reach full maturation and be able to ovulate. For the oocyte meiosis, we also didn't see any difference. We don't see any difference on the oocyte size. And also for the post-ovulatory P4 from the corpus lutein, we also don't see a lot of difference. They even a slight a decrease, but not significant. But next, we also want to see whether we can see other defects, because we only choose some genes to do the qPCR, but we also want to know whether there are some other defects. So what we did is we just sequenced those follicles with different concentration of the FSH, and what we found is, so actually compared to mild FSH, the excessive FSH, they can cause inflammation in the ovary. So you may want to think about, ovulation, inflammation is fine because ovulation is an inflammation process. But actually for here, I want to highlight, actually this ovulation, this inflammation, it happened before the RH surge. So that's not good. For example, PCOS is an inflammatory disease. If before RH30, if the follicle or the ovary is inflamed, that may not good. Either increase the risk of the ovarian cancer, or it's not good for the follicle or oocyte quality. So for example, we see a big induction of some like ovulation specific markers and also a lot of inflammatory genes. And we also measure the like a media and see how much of the prostaglandin is there. Actually, we also see a significant increase of the prostaglandin. You know, prostaglandin is not good for the ovary. Only good for ovulation, but they need to drop. Otherwise, it will do some bad things, especially the increased risk of the ovarian cancer. So that is what we found, but we don't know whether it happens in human as well. So that's what we rely on like the people in this room. So, and then we also want to understand how about the oocyte quality? Because for hormones, maybe not too bad, right? Because we want to make sure the egg is really in good quality and normal, and the ovarian stimulation doesn't affect the egg quality. So overall, the answer is yes. So we did the SMART-Sync single oocyte RNA sequencing at the transcriptome level. They are okay. So, and they are very highly like correlated between the male and the high level of the FSH. But interestingly, we found a lot of like for the differentially expressed genes, most of them they are downregulated. So high FSH will downregulate a lot of genes in the oocyte, and most of them are related to mitochondrial and metabolism. So what we want to claim safety probably not very significant. We don't know what is the like whether they really affect the mitochondrial function or the metabolism in the oocyte. We still don't know the answer. Yeah. So overall, that is what we found from this article we recently published in Endocrinology. So this FSH threshold required for follicle to become dominant and maintain the maturation, and it goes through the whole process to become ready for ovulation. And the high level of the FSH, they can induce high level of the estradiol. We already know, progesterone we already know, but strangely also testosterone. And also high level of the FSH, they may induce inflammation in the ovary, especially in the granulosa cells and maybe theca cell too. And we want to ask whether the mild or individualized ovarian stimulation might be better for a patient. But that's like it's not my focus, but we can discuss during the dinner time. So for the next part, I'll just very quickly go through the PCOS, just because I heard most of the patients coming to IVF clinic because they don't ovulate, they have PCOS, so they are infertile, right? So we just use this model to mimic whether we can mimic the PCOS ovary. So I think I don't need to introduce this letter. We know PCOS very well. It's a very complex disease. We don't know the etiology very well. So what we want to do is we want to mimic the like abnormal GnRH gonadotropin secretion pattern. We give them high RH, but we use HCG here, and relative to the FSH. And then we want to see whether we can mimic what is happening in the ovary in PCOS patient. So that is our recipe. We give them different ratio is to see the dose response. And then what we found is whenever we change the gonadotropin ratio, we can see a lot of testosterone production. Okay, that's good. And we already have 2 papers demonstrating that. And we also see actually they don't ovulate like very well. We see a dose dependent effect on the ovulation reduction too. We see right here. And for the oocyte, we don't see very much change actually. So as long as the follicle can rupture, we can see the powder body. So that means oocyte probably less sensitive. And for here, I have just 2 quick videos to show you what the PCOS will look like. And this is the control video I already showed you. And at the bottom is the follicle with PCOS, what we call the PCOS follicle. You see actually the control follicle, they can rupture pretty well. You can get metaphase oocyte. But the PCOS follicle, I think it bounced in there, it tried best, but it didn't rupture, for some reason. And then we also did sequencing, using granulosa cells, and also we use oocyte too, but we don't have data for here today. And we see actually at transcriptome level, actually a lot of genes are changing in PCOS follicle, and including some marker genes. The SILK1, VCM1, there are some, like if you check the NIH reporter, a lot of scientists, they focus on their function in PCOS. And we also, PCOS, everybody may already know, PCOS is an inflammatory disease, including in the ovary. We also see a lot of induction of the PTGS2, probably PTGS patient, they have prostaglandin E2 and interleukin 6 in the follicles, and also the interleukin like 1B. And so for the what kind of plot right here, so we summarize the top like upregulated genes. So currently our like a way is we want to see whether some of them they are druggable target, whether they are the key driver of the PCOS, and then we can give them antibody or small molecule inhibitor, and then the PCOS patient they may can like ovulate better. And if you want to do the IBM in vitro maturation, so whether we can add that stuff in the culture media to have them better like to have better like a maturation outcome, and also at the same time maintain the oocyte quality. Yeah, so that is what we are doing, and what we want to do in the next 3 years too. So for the next part is that we talk about the ovary, right? Like everything is about the ovary. And again, we are also interested in the uterus because we want to make the ovary uterus on chip to study, make it more complex and also human relevant. So we just use the same idea, like a hydrogel encapsulation method, but we collect the human endometrial and stroma cell where compared to the ovary is relatively easy. So we still do the 3D culture, and we want to induce the desaturating process to mimic the uterine receptivity and the embryo punctation. Now, overall, the answer is yes. And we can use estradiol, cyclo-MP, and also progesterone EPC is very well accepted in the field. You can get this like a lot of paper using EPC. But our idea is whether we can co-culture the ovary and the uterus and make it happen too, because that is what happening naturally in vivo, right? So you see our data indicate, so compared to the EPC, actually, if we culture the uterine endometrium organoid, yeah. So actually, if we co-culture the endometrium organoid with the ovary, we can also induce the desaturating process too, based on the like the swelling of the like the endometrium cell that are human, and also based on like some marker genes, for example, the IGF-BP1 and also the prolactin 2. So we have a lot more data, but these are unpublished, so we are still working on the paper, and due to the time, I don't have time to show it. For example, we also did sequencing and other in-depth analysis, and we also use this model for toxicology research too. So our like long-term goal is we want to build a really human cell-based female reproductive system on chip to study the IVF, reproductive disease, and also toxicology as well. So yeah, again, this is our project. If you are interested, feel free to email me because I primarily focus on the like on chip and organoid culture. But if you want to study how environmental change, like climate change, and also extreme heat and other factors will affect the like IVF like a success rate, I'm open to collaborate too. And if you are doing oncofertility and you are interested in the fertile like a protected drug development, we have several very good targets, but there's a gap between the academic professor and the big pharma company. Johnson Johnson and BMS, they are not interested in our research because they think it's too early. Yeah, so and last but not least, I would like to thank like all my collaborators. You see, I didn't do any of the work, but all rely on my postdoc, graduate student, lab technician, and our collaborators too. Of course, I want to thank the funding support, and I have two pigs right here, dolphin and female soldier, because we got a DoD grant to study how PFAS will affect female military ovary health. Yeah, actually, they visit a lot in their clinic too. And we used to make a dolphin ovary on chip to study how oil spill and climate change will affect whale and dolphin ovary, and whether you consider to do the IVF for dolphin, maybe, right? A lot of IVF for panda in China. So yeah, with that, I would like to thank for the invitation again. I really enjoyed the Taylor time, the conversation, and I look forward to the discussion and also the dinner time for further discussion. Thank you.
Can I ask the first question? It was your paper on the FSH ceiling that made me aware of what you're doing these days. We are very interested in age-specific ovarian function. Does this change? Does the ceiling change? Does the amount of FSH changes as in your models, animals are getting older?
Yeah, I believe the answer is yes. We didn't compare different age, but based on my understanding, for example, some research found for aging people, their FSH is more glycosylated, and then it's not active as the less glycose, like a younger, the FSH from younger women. And also in the ovary is more fibrotic, and that will make them less responsive to FSH. So actually, this idea is easy to test in our model. We just need to collect the follicle from young and aged mice and give them either different glycosylated FSH and see what is happening there, including the OSAT too.
Why isn't anybody doing this? I mean, the glycosylation data have been known for
Yeah,
30 years.
Yeah, probably sometimes it might be a old school concept, and then people think it might be good for, it might not be like novel enough to get a grant from NIH. Maybe that's the reason, I guess. But I know Dr. Raj Kumar from Colorado and also Dr. John Davis from UNMC, Nebraska Medical Center. They are doing the glycosylation of the FSH. I'm happy to make the connection too. And the reproductive agents were a hot topic too. And I believe that if we combine both parts, that would be pretty like a significant research for us to do and also advance the IVF clinic.
Anybody else?
Thank you for the very interesting thing. I have a couple of questions about the human follicle organoids. They were so inefficient in giving rise to oocyte. And I'm wondering, did you test a range of conditions? Because I would say the big question is what the alginate embedding is doing to the organoid. And did you try different embeddings with different functionalization from, ligands, cell addition molecules, stiffness of the embedding? And is this going to be relevant for improving the condition for the human side?
Yeah, that's a very good question. Actually, in mouse follicle, we try. For example, if we change make estrogen concentration higher, it mimic more about the PCS follicle. The stiffness, the more stiff, like a more rigid environment will make them secrete more androgen. So that can mimic the aging ovary. And that's primarily based on the mouse follicle. And some bioengineering friends, like we collaborate, they also make a degradable alginate to make it mimic more like an in vivo. And regarding human follicle, we come back to the tissue limitation topic again. It's just so hard for us to get a premenopause ovary. And even for the 56 follicle, I accumulate them for within 3 years. And for human, every research when they come to human, the variant is so big. The human is so much more complex than like a mouse in the lab or in the animal room. And then the variant is so big, and we cannot make any conclusion. Yeah.
Did you say that androgens rise as the animal gets older?
Yeah. Stiffness. I don't know whether the aged animal will have more androgen in the blood, but if we change the stiffness of the ovary, they will produce more androgen.
That is interesting because in older women, androgens drop.
Mm-hmm. Yeah, I think the androgen drop primarily systemically. But for example, for 2 aged women, like if one, I believe for one more like a rigid ovary, they will produce more androgen compared to the same like older age of the women with relatively softer ovary. But if we compare to young women, because their ovary is not that functional, so they have relatively lower androgen.
Just more by the follow-up question. So for the endometrial organoids, so are you planning to combine stromal cells plus epithelial cells? And what about the spatial organization of the two? Do you get something that is stratified in the right way to look like an endometrium? Yeah. Or you get salt and pepper?
Sure. Well, very good question again. So we currently, this model or the data I showed today, they don't have epithelial cell there. But for example, Dr. Franco D'Amaio from NIEHS, they have very fancy research regarding like actually epithelium is very important for inducing desegregation. So that might be the reason we still need to use a little bit cyclin P. in our media. Maybe cyclic AMP is like induced by a ligand in the epithelium or the signal in the epithelium. But that's our plan. But the challenging part is for uterine epithelium, whenever you get it out, and then they differentiate, they are not epithelial cell anymore. So that is the challenging part. They are not as stable as stroma cell. For stroma cell, they are not primary. They were primary, but you can even passage them as long as the generation less than 10, they're still okay.
My question also has to do with the how much you can translate this work to humans. You have the human egg, the gene expression patterns are similar, but they're also significantly different compared to mice, for instance. And then there's some data which suggests that, because of the time scale, there are some like epigenetic changes that also happen in the human egg compared to the mouse egg. So in terms of like translating, have you thought about moving to like primate models or something that's more accessible compared to a human ovary?
Yeah, so the answer is I really want to do it. And as a university professor, the results has a really like a limitation. For example, we collaborate with Mary Zaninsky or Oregon Non-Human Primate Research Center a lot, so we can pass on the information so they can do it. And for human, I'm always I always welcome REI collaborators so we can translate our finding to using human samples. For example, if some of you you are interested in the high androgen, I really want to know whether ovarian stimulation will induce the androgen in their blood or follicular fluid. I asked around, nobody has ever measured that before. But androgen is not, testosterone at least, it's not that hard to measure, right? Yeah, it's a routine, but after ovarian stimulation, probably, maybe you have, but I asked several REI, they don't measure the androgen. And then it may change the concept of premature luteinization. I don't know the answer is yes or no. So that's so I claim the species different is a big issue actually. But unfortunately, the stem cell induced ovarian cell, including germ cell and somatic cell, or the organoid, it is really not ready for a research as long as you focus on stem cell.
Thank you for the presentation. I have some theoretical questions.
Sure.
I think that ovarian chip is an exciting project. And I wonder if there is any delay, if there is any possibility to delay the ovarian aging by decreasing the monthly development of the number of follicles in the ovarian.
Yeah, I think that's very possible. If whether it's like a pro and cons, whether if we delay the follicle maturation process, but they will miss their they will don't have their cycle too, and also progesterone is protective for uterus, and whether they will have high risk of endometrial cancer or ovarian cancer, we don't know. Yeah, but that's possible. And also some people they found melatonin and other antioxidants they can delay the ovarian aging, but in mice again they're not. In human that take years to study that. Yeah. But very cool idea.
Following up on my colleague's question here, the two aspects that we already heard, the control of the microenvironment for the organoids, the level of stiffness, and how do we measure what is the optimum conditions for that, knowing obviously that inflammation is involved, so stiffness will change in function of time. But also curious about the timing of events. As you mentioned, obviously, a cycle for a mouse is totally different than a cycle for humans. And maybe there are evolutionary reasons, maybe there are other reasons. But have you tried to figure out what regulates timing as opposed to physiological processes? For example, if you were to make a chimeric organoid, where it would be half mouse, half human, would it develop at the rhythm of the mouse, or would it develop at the rhythm of human? And regardless of the answer, why do you think that is?
Yeah, that's a cool idea. I didn't know the answer. Yeah, there are some humanized mouse model, but for follicle, I used to culture follicle for more than 10 days, keep them FIC, and then they are aged. So that means, for some evolution reason, or maybe the genome, epigenome, the epigenetic regulation, that's the beauty of the nature. So they only need about 450 days for the whole process in mice, and then 200 days for the whole process in human. But that's a cool idea. Yeah.
For example, so if there was a autonomous effect, you would think that if you make a mixture of 50-50, each one would develop on their own rhythm.
That's
But if
very
it was a
possible.
non-autonomous effect, then one would take over the timing of the other.
Mhm.
I think that is something I'm very curious about, the control of time. The
Yeah.
process seems to be similar with species-specific attributes, as you mentioned. But what really controls time?
Yeah. So currently, I expect the genome and epigenome, those like a control signal pathway primarily regulate this process. They just need 60 days during the like 100,000 years of the evolution. Yeah. That's what I expect.
Do you think it can accelerate the human program?
For the good question. I don't know the answer. Yeah. But it's a very good direction to go actually.
Yes.
Yeah.
Okay, time to eat.
Okay, thank you so much.
Thank you very
Thank you
much.
for
You can find your way down the stairs, and we are eating right next door at the Shalom. Or you can squeeze into the little elevator and have it go up and down a few times. Okay?
Public question,